right, guys, thank you for coming out today for our Knauss Brown Bag. I am excited to introduce Katharine Egan. She is a Knauss Fellow this year, and she's working as an Ocean Exploration Fellow in the NOAA Office of Ocean Exploration and Research. Today, she is presenting to us. She recently graduated from the University of the Virgin Islands with a MS in Marine and Environmental Science, where her thesis was focused on predicting coral distributions. Prior to the Knauss Fellowship, she worked as a GIS Analyst for the NOAA National Centers for Coastal Ocean Science in Beaufort, North Carolina, providing project support for spatial predictive modeling of threatened ESA corals, the National Coral Reef Monitoring Program, and the assessment of hurricane impacts to coral reefs in Puerto Rico. Katharine has also completed an internship with NASA related to disaster prevention and has received funding to prevent marine debris through outreach in the US Virgin Islands. Today, she will be presenting her master's work, predicting the distribution of threatened Orbicellid corals in shallow and mesophotic reef ecosystems along the Puerto Rican shelf. Thank you, Katharine.
Thank you so much, Hollis. And thank you so much, everybody, for coming out today to listen to me present the results of my thesis research. I'm really excited to be here. This is a NOAA-funded project, so it feels really good to be able to present it at NOAA in Silver Spring. So I'm going to start by providing you all with some context on where my thesis research took place. It took place along the Puerto Rican shelf, which is at the intersection of the Atlantic and the Caribbean Sea. And here's the shelf area, which of course encompasses the big island of Puerto Rico, as well as Culebra and Vieques, which are part of Puerto Rico, St. Thomas and St. John of the US Virgin Islands, and the British Virgin Islands. So St. Croix, which is part of the USVI, and Mona, which is part of Puerto Rico, are on their own shelf edges. So this is the shelf edge that I'll be focusing on today. And this shelf area contains a lot of unique coral reef and hard bottom habitat, which you can see pictured up here at the top. However, something that this benthic habitat map is missing generally are mesophotic coral ecosystems, which are reefs that occur in areas between 30 and 150 meters. So they are deep reefs. And because of their depth limit, they're considered low light environments. However, they're still warm water habitat. Because of the warm water component to mesophotic reefs, they support a variety of corals, sponges, algae, and commercially important fish species like this Nassau grouper pictured here at the top. And something that makes mesophotic reefs unique compared to shallow reefs is that they have a really high coral cover, particularly in the upper mesophotic zone. So one of the coral genera that compose the cover in mesophotic ecosystems is Orbicella. And so I have all three Orbicella species pictures listed up here. So they're abundant in mesophotic reefs compared to shallow reefs, but they're an important reef-building species throughout all depth ranges in the Caribbean. And something that's really interesting to note is that they are actually listed as threatened under the US Endangered Species Act as of 2014. So Orbicella fabulata, pictured here in the middle, and Franksi, pictured on the bottom, are the ones that are most abundant in mesophotic reef ecosystems. Annularis tends to occur in areas that are more shallow, so they're very rarely found at mesophotic depths. And pictured here, I also have the shallow water morphology of these two coral species, because at mesophotic depths, they tend to adopt this more plate-like or planar morphology. And this is so that the colony can maximize the amount of light that reaches it. And so this creates a very unique looking reef ecosystem with a very unique looking rugosity as well. So there are a number of environmental drivers or variables that describe the current Orbicellid distributions. So I already highlighted that depth describes Orbicellid distributions because they're more abundant at mesophotic depths. But there are other drivers as well, such as temperature. So temperatures at mesophotic depths are much cooler than at shallower depth limits. And then also things like seafloor topography, so the rugosity and slope of an area, and the availability of hard bottom habitat are also determining factors for Orbicellid locations within these regions. And another very large environmental driver for Orbicellid distributions is also wave exposure. So pictured here, I have a schematic from Dunn's paper in 1983 showing the zonation of some coral species based on some environmental drivers. And we could see that Montastria annularis or Orbicella annularis occurs in areas with much lower wave energy. So Orbicellids prefer sheltered reef habitat. And that can be seen up here on the top schematic, where wave action decreases as you move offshore into mesophotic depths. So mesophotic reefs are generally sheltered habitat compared to shallower reefs. But despite what we know about Orbicellids right now at mesophotic depths, there is a lot of information we don't know about mesophotic reefs in general. And that's because they're difficult to study due to the depth range. So a lot of people characterize reefs in these areas using ROVs or drop cameras, as well as folks who have technical scuba diving certifications. However, these methods can be limiting when trying to describe the spatial extent and location of mesophotic reef ecosystems. And this was highlighted in a paper by Locker et al. In 2010, in which the researchers focused on identifying areas of potential mesophotic reef habitat using only depth. So they didn't really look at anything like hard bottom habitat or areas where there was a high abundance of coral, because that kind of information wasn't available when they were creating these maps. So you can see that here in the yellow area. And they highlighted how important it was to identify actual areas of high cover mesophotic coral ecosystems. So my thesis research aims to address this problem. So I aim to identify the locations of Orbicellid species using variables that drive their current distributions. And in turn, we could find areas of unexplored mesophotic reef habitat along a portion of the Puerto Rican shelf, while simultaneously creating habitat maps for these threatened coral species. And also I was interested in seeing how the abiotic variables contributed to identifying both Orbicella and in turn mesophotic reef locations. And the method I used was species distribution modeling. So I created 2 species distribution models for 2 coral groups. First, I did annularis, which I said is not abundant in mesophotic reef ecosystems. It's rarely found there. But I did a model for it anyway because I wanted to see how the model would perform using Orbicella annularis to see if it would be predicted within those shallower areas. If it predicted at mesophotic depths, then there's probably something wrong with the model. And then I conducted a model using a combination of Fabulata and Frankside. And I combined them together because these 2 coral species are really hard to differentiate from each other at mesophotic depths. So it just made most sense to put them together because they occupy the same niche habitat. So I have a little schematic of how you do a species distribution model for you all, and I'll move through it as I move through the rest of my presentation. So before even conducting any sort of model, it's really important to gather the data that are needed for the model. So here is the extent of my model in the outlined gray area. It encompasses an area of about 3,500 square kilometers, and I cut it off at a depth of 60 meters. It looks like this because it was based on the available bathymetry data that I have for the region, so it's kind of got a weird shape. I also wanted to show you all the transition from shallow depths in light blue to mesophotic depths. It's not necessarily a hard cutoff, but I just wanted to show where generally shallow areas occur versus mesophotic areas in this model extent. And here are the distribution of the occurrences that I compiled for this project. I compiled Orbicella presences and absences from NOAA-funded projects, as well as a number of projects conducted at UVI. I have them listed here. So in yellow, we have Fabulata and Frankside presences. Annularis is in green, and then the presence of both coral groups are in blue, and then I have absences in the dark gray circles. I tested 22 abiotic predictors, but 8 ended up creating the best model for both coral groups. And so that spanned across things like currents, temperature, wave exposure, seafloor rugosity, or seafloor topography, and depth. I also sampled up or down, depending on what resolution the predictors were at, to a 50-meter resolution, so the resulting model came out at a 50-meter resolution as well. So for the statistical model I used maximum entropy, which is a Maxent, or called Maxent model, which is a presence-only or background sampling species distribution model. I'm not going to get into the parameters that I used for it, but I'm happy to discuss with anybody later if they really want to know. The spatial prediction output from Maxent is a continuous grid where each grid cell represents the probability of that species occurring within the grid cell. And so that was on a scale of 0 to 100%. And so I also applied a threshold to that continuous grid to create a binary grid, which shows the differences, which will show the presence of the species being absent or present in a particular area. And I tested the model performance in a number of different ways. I used the discrimination capacity, which I measured using area under the receiver operating curve, or the AUC value. I also used this interesting method called reliability. And so this was the agreement between that spatial prediction output and the presences and true absences. So what I did was I took both the presence and the absence data and I overlaid it onto the continuous spatial predictions. I subtracted those values from each other. I took an absolute value from all of those values, and then I averaged it together to get one value, and that's called the mean absolute error. So when the mean absolute error is very low, it means that your absences and your presences are generally agreeable with the spatial predictions, and a higher MAE means there's disagreeability. And I also measured the uncertainty. So I conducted 10 replicate model runs for each coral group, and I took the standard error of those spatial predictions and averaged across them. And like I said in the beginning, I also wanted to look at abiotic, the contribution of the abiotic variables as well. So Maxent gives a % contribution of each abiotic variable and how much it contributes to the final predictions, as well as information on response curves. So how each variable, which is on the x-axis, changes with the prediction, which is on the y-axis. All right, now for the results. So here at the top is the spatial prediction output for Orbicella annularis. So areas that are in more yellow to red are indicative of a higher probability of annularis occurrence. And we can see that generally it occurred in shallower areas around the islands of St. Thomas, St. John, Culebra, and Vieques, and generally did not really occur at those mesophotic depths, which you can see on the southern insular shelf. And this is highlighted when we applied the threshold, so those green areas are a probability of annularis being present, which is generally shallower areas. And overall, the model performed quite well. The AUC value was pretty high, so the model was excellent to outstanding. There was also low variability between the model runs, and that MAE value was at 15%, which is pretty low, so the presences and the true absences were generally agreeable with the final spatial predictions. So this indicated to me that the model was performing quite well. So then I conducted the models again on Fabulata and Frankside combined, and we can really see how Fabulata and Frankside would act as a good proxy for where mesophotic reefs are located. There is a big area here between Culebra and St. Thomas called the Virgin Passage, where there's a very high probability of Fabulata and Frankside occurring. And there's a lot of areas along the southern insular shelf too that are highlighted within this model. And then once again, when we apply the threshold, we can really see where those areas are located, where the probability of them being present are located. The model was still, the model performance was still quite good. The model was excellent to outstanding according to the AUC value. There was generally low variability between the model runs. However, the MAE value was a little bit higher compared to annularis, so that suggested to me that there was some disagreeability with the predictions. So what I did was I broke up the absence data and I averaged that together, and I broke up the presence data and I averaged those together to compare the two and see where it went wrong. So the absence MAE was much higher than when you average all of the values together. So that suggested to me that the model was over-predicting. It was predicting into regions where we have recorded absences. And so it's important to take into consideration that absence data can be quite unreliable. It's a big debate amongst people who conduct species distribution models on what kinds of models work best, presence only versus presence absence. And a lot of people make the argument that absence data is unreliable, so a presence only model is a good way to go. So generally, for the MAE, absence data might be unreliable in this situation. I'd have to look into it further. However, the presence MAE was still quite high, so that indicated to me that there were incorrect predictions in some regions. And there are a number of reasons why this could be happening. First, there are missing or inaccurate abiotic predictors. So something I didn't have in this model was a benthic habitat map. So my models do predict over soft sediment, and corals, of course, do not occur on soft sediment. So I'd have to go back and check those areas to see if the model is predicting into those regions. Additionally, these models are not capturing fine-scale abiotic processes, biotic processes, or any anthropogenic stressors, which we know can affect the distributions of corals within these regions. However, I think my models are a really good starting point, and I think it's important to continually update them as new data become available. All right, and so for the variable importance, it turned out that regosity, depth, and sea surface temperature range were the three predictors that contributed most to the annularis predictions. And I had those response curves shown right up here, with regosity contributing more so than depth and the sea surface temperature range. And what you can take away from these graphs is that annularis preferred areas of higher relief habitat, so regosity greater than 1. It also preferred shallower waters, which we knew already. And it also preferred more stable temperature ranges. So occurrence decreased as sea surface temperature range increased. The results were similar for Fabulata and Franksci, with regosity being the top predictor, and wave direction and average sea surface temperature coming in third. And a similar situation with the response curves. Fabulata and Franksci preferred more higher relief habitat. The response curves indicated a peak at about 85 degrees wave direction, which is actually indicative of the southern insular shelf wave environment. And I think that was just due to the concentration of presences that occurred within that area. But I'll discuss more of that in a second. And also there was a peak at about 27.7 degrees on this response curve. So it preferred maybe has a prime temperature range in this scenario. So why was regosity one of the top predictors for both coral groups? First, I want to just explain how I calculated regosity in this sense. So regosity was a measurement of the surface area ratio that was calculated from the bathymetry data. So it's not really a measure of any sort of fine scale regosity or structural complexity. It's more like a relief measurement. And it's unitless. So anything greater than 1 is indicative of high relief habitat, and anything less than that is indicative of low relief habitat. And I know what you're all thinking. Corals do create regosity. I totally get that. And we can see that here from Graham and Nash's paper from 2013. So with an increase in structural complexity, there's an increase in coral cover. And we can see this also with the branching coral cover too. However, I think with these models, it's important to consider the spatial scale at which they were conducted, which was at a 50-meter resolution. So I don't think the corals were creating all of the regosity at that spatial scale. It could have been something else. And I think what was causing that ideal habitat were drowned reefs in the region. So modern mesophotic reefs are built on antecedent reef structure. And these reefs were reefs that occurred during the Pleistocene, but became drowned reefs when sea level rise as we move through geological time. So I think those drowned reefs are what's providing the substrate and regosity for the Orbicellids to settle and grow on. Additionally, regosity could be acting as a proxy for another predictor. It's highly correlated with slope of the sea floor. And slope was not included in the model because we don't include correlated predictors in models. But the slope can affect things like sedimentation, which in turn can affect Orbicellid distributions. It can also affect coral recruitment and settlement. Coral recruits prefer that vertical surfaces and upward facing habitats, which can affect the distributions in turn. And then I wanted to discuss wave direction as well. I found wave direction to be a little bit puzzling. I think that it's acting as a proxy for another variable similar to regosity. And I think it might be acting as a proxy for the underlying topography of the region. So the mesophotic reefs in this region are a spur and groove habitat. And along portions of the Puerto Rican shelf, they are oriented to the, they're parallel to the wave direction. So in a southeast to northwest direction. And so I think the model might just be picking up on the underlying topography in these regions. Additionally, I found a study that was conducted in Hawaii, which was looking at coral cover as it was described by a number of environmental factors, including things like wave direction. And they had found that wave direction from a certain region, so from this region that I have up here in the circle, there was a lot of limited coral cover in this area that they found correlated with wave direction, or they found that was described by the wave direction in that area. And so they found coral cover to be low, and they also found that wave direction was correlated with wave height in this scenario. So they suggested that wave height was actually contributing to the low coral recruitment and cover within this region. So I'm wondering if something similar could also be happening along the Puerto Rican shelf, and I'm still actively working to figure this out. So to conclude, now have maps of potentially unexplored reef along the Puerto Rican shelf that can also couple as habitat maps for these threatened coral species. However, as I stated earlier, it's important that we continue to refine these maps as new data become available. And once we can refine them more, I think it's really important that they are ground truth to see if there are actually unexplored reef or reef in the areas in which the models are predicting. And finally, I plan on making the spatial predictions publicly available through NOAA's NCEI after I publish the paper. So if somebody wants to ground truth them, they can just access them and do it themselves. So let me know if you do that, though. And with that, there's so many people that I want to thank for helping me on this project. I worked under the discretion of Tyler Smith, who is my advisor at UVI, as well as my committee, which composed of Shay Beeman from NOAA NCOS and Dan Holstein from Louisiana State University. Thank you to the Brown Bag Committee for putting this on, and for NOAA Library for hosting me today. And with that, take any questions. Yeah.
Great talk and great work.
Thanks.
I'm curious if any of the early predictors for your model included zooplankton density, because we know that sometimes at those mesophotic depths, coral heterotrophy plays a greater role just due to the fact that they're not in that photic zone, or still in the photic zone, but maybe not relying as much on light. Just out of curiosity, were any of the early biotic predictors zooplankton density?
Yeah, it's a good question. Oh, yeah, sorry. So the question was, did I use any zooplankton density or predictors in earlier rounds of the models? And the answer to that was no, I did not. I didn't actually look at any biotic predictors for this model at all. There was some talk about using chlorophyll A, which I know is not zooplankton, but I had read in the literature it wasn't necessarily a great predictor for Orbicellids, but definitely something to include in the future.
Yeah,
that'd be great. Yeah. Yeah.
I was actually something similar to that. There's a few papers that I can send to you that talk a lot about wave height and wave direction and predicting benthic communities. And part of that also has to do with different wave directions being influenced internal tides, which is used as well in events on reefs.
Wonderful. Yeah, I'm gonna just read that to say that to the people. So someone is kindly is going to kindly send me some papers about wave direction and how it describes coral communities. Thank you, Amanda. That would be awesome. Great. Yeah, that's what I'm kind of struggling with the most on this right now.
I actually have a friend who is the author on a few of them who works for NOAA in Hawaii. I'm sure we have each other.
Perfect. Thank you.
Yeah.
Anybody else have any questions in the audience? Did you want to read their questions here?
So yeah, our online question, did you include dead standing colonies in the presence data?
Oh, no, I did not. So I actually looked at, so the data that I compiled from had information about the percent cover of live coral. And so I always made sure to include any colonies that were for sure there and any most recent data that I possibly could have for the region as well. So assuming they're still there. Are there any questions here? Yeah.
So these deep guys, what is the biggest threat for them? Is it are they getting as warm? Are they as likely to bleach, or is it more of a sedimentation?
Yeah, that's a good question. So the question was, what threats are occurring at mesophotic depths and on mesophotic reefs? I would say bleaching is a really big concern. So actually, this work was conducted by my advisor in the Virgin Islands, but him, amongst many other researchers, found that corals at those depths have a lower bleaching threshold, so they bleach at lower temperatures compared to shallower reefs. So they're actually more, I would say, maybe in danger than shallower reefs are in that regard. There's also problems with sedimentation, and actually some work that was conducted out of the University of Puerto Rico, Mayagüez, had also found a lot of anthropogenic disturbance on mesophotic reefs as well in sections along the Puerto Rican shelf. So they experience their own host of threats just like shallow reefs do.
Okay, even though they're probably not relying on the zooplankton for nutrients as much, because can they photosynthesize?
Yeah, yeah. These are photosynthetic corals. Yeah.
And then another question is, how would your model translate to other mesophotic areas outside of Puerto Rico?
Yeah, that's a good question. We're looking at that right now, actually. So the problem is lack of available data. We had talked about translating this into areas that don't have nice high-resolution bathymetry data like we do, but the problem always was, how do we get those kinds of data? And I think some folks at the University of the Virgin Islands are working on this right now, working on trying to get depth data for regions that doesn't have access to the technology that we do to collect those kinds of depth data. Because I think once you get your depth data, you can get your seafloor topography data, and then I just got sea surface temperature data from NASA, and there are options to get wave exposure data for these regions as well. So depth is our biggest limiting factor, but it would be great to translate this to other regions for sure. Yeah.
Along those same lines, I think, don't quote me on this, but Flower Garden Banks has really extensive benthic maps. So like one place that you could test your model would be there, because it's my understanding we have good data in that area. So,
yeah, that's good to know. Yeah, for sure.
A ton of free time right now, but,
so the comment was, I could test my model in Flower Garden Banks because there's extensive data in those locations. Yeah, definitely, that's totally an option. And we know Flower Garden Banks has a ton of Orbicellid in it, so it'd be a good test ground for that. Yeah, it's a good question. It's a good idea. Yeah.
If you were going to translate this, I was thinking about that, like why it would be a great place for you to test it. My lab has actually done a lot of like rebreather mapping and mesophotic reefs out there.
Oh, nice.
If you were going to translate this and you were going to pick a specific coral species to sort of have near Orbicellids, do you know what one you would pick?
So the question was, if I could pick a Pacific coral species to conduct these models on, which would it be? I don't know any Pacific coral species. Yeah, I've never even seen a Pacific reef. I've done all my work always in the Caribbean. Yeah, I don't know. Exactly. Yeah. No, that's a good question. But I mean, I did draw on a lot of papers. Like I drew on work that Brian Costa and co had conducted in the humpback whale national marine sanctuary in Hawaii, because they used a MaxEnt model on a number of different coral genuses out there. So it's possible and can be translated in those regions for sure. Mm-hmm.
This is 2016 paper, my advisor's paper, is all.
Okay, perfect. Yeah. You gotta send those to me. I need them.
Yeah, well, thank you, Katharine.
Yeah,
We're just going to take a quick break before the next presentation, and we'll forward the remaining online questions to you.
All right, sounds good. Thank you so much, everybody.